I think we're saying over and over the same thing. So yes, let's say that this drug is preventing MER33 from doing what it normally does. So it's blocking it, hindering it, right? So, but how do you explain that at higher doses, you have less and less and less MER33? Oh, there you go. So not getting triggered. Okay, I like that. So can you elaborate a little more? But does it trigger here at higher doses? No. I think you're just flipping it. So what you're saying is it may not be triggering the mechanism at 200, and it triggers. I like that. Yeah, correct. And that's one of the reasons I wanted to bring it up is that happens a lot in these molecular / genetic networks, that you have certain effects that don't appear until higher dosage of the gene, the protein, the drug, the abiotic variable like the temperature, like the pH, and so forth. So there are many things that may not kick in until a certain threshold. And it is absolutely conceivable, and that's sort of the idea that the authors go with. Actually, I have the language that they use. They say these ASOs, the oligonucleotides, can block microRNA function promoting microRNA degradation by engaging RNase-H or by preventing its access to target mRNAs. And I think that the underlying idea here is that at lower doses, the oligonucleotides will prevent access of the microRNA to the messenger RNAs, in this case the ABCA1 messenger RNA, and at higher doses, it will, using your language, it will let the RNase-H mechanism kick in and destroy the MER33. You had a question or comment before?
I was curious if you knew what the mechanism was for the ASO against when it is given at higher levels, why it does degrade MR or MER33, like if it's a cleavage.
Yeah, that I'm gonna make so the objective, honest answer is I don't know. My best educated guess is that it's just a matter of, in a way, sensitivity to a signal. So what happens is what we know that these ASOs bind to MER33 by base complementarity because they're just reverse complementary to them. My educated guess is that RNase-H may be patrolling the cytoplasm, right? And if you have a few ASOs binding to MER33s, that's enough to prevent each of them from finding the mRNA and doing their job. But it may not be enough to bump into RNase-H and wake it up of sorts. Does that make sense? So it may simply be a matter of thresholds of sensitivity to the presence of these things. So MER33 is super sensitive to the presence of ASO. Why? Because it's in its very nature to be bound by the oligonucleotide. Whereas activation of RNase-H depends on the bumping of these complexes onto them. Does that make sense? All right, I want to make sure that I get... Cool. So the authors think and argue that this may be why, even at a lower dose in which you're not triggering destruction of the MER33, you're still inhibiting the translation of, you're inhibiting the function of the MER33. You're not destroying it, but you're still inhibiting it, and that's why there is an upregulation of ABCA1. But I like to go now. I know that this probably wasn't exactly what you had in mind, and at first I hesitated, but now I'm like, let's go with that idea. So, fine. Perhaps the MER33 ASOs target something else. So it's not so much about what they do to MER33. It's about you said the transcription factors that control them. A little hard to imagine because an oligonucleotide doesn't really have a very direct way of interacting with that transcription factor. But maybe this oligonucleotide is targeting some other microRNA that in turn controls the activity of the transcription factor. So the point is, even though what your idea was saying is not exactly this, we can play with that. In addition to the argument that the authors made, which is valid, as usual, we need to play devil's advocates with our own data, right? So that's what scientists do. So I could imagine if you're in the lab facing these results, and if you really want to be as harsh as you can with yourself so that you can move forward with confidence with your conclusions and your results, one of the things that you could argue is, well, perhaps these miR33 oligonucleotides are targeting something else that inhibit ABCA1 expression not via, so via some other mechanism, in a manner that is miR33 independent. That might be an alternative explanation to the same results. Someone shows me results that say, look, I treat these cells with this ASO, and I don't see any changes in miR33 levels at 200 nanomolar, but I do see changes in ABCA1 expression. Can you please explain that to me? One argument might be, oh, don't worry, these ASOs don't always degrade the target mRNA or the microRNA, so that's why you don't see any changes in microRNA, and yet you see changes in protein expression. An alternative might be, well, maybe it's working through some other mechanism that has nothing to do with miR33. We're all on the same page about that one? Yeah? How can we test that? With what? I'm having a hard time. Silence the. Okay, so and tell me what the experimental design would be. You have your cells, you silence miR33, and what is it that you expect to see?
Well, if the ASOs do target something else, then there would be an inhibition of the ABCA1. But if they don't target something else.
Remember that, so what we have here, so when we add ASO, ABCA1 goes up, okay? What the authors think is that what ASO is doing, so there is miR33 inhibiting ABCA1, and what the authors think is that the ASO is inhibiting miR33. But now what you're saying is, oh, what we can do is take out miR33. So we do that. How will that help you in determining if the ASO is working through miR33 or working through some sort of independent pathway?
So if we don't have
The target.
the target and it's still inhibiting.
Oh, I see. So I think I got your idea, and we'll discuss it further. Yes?
Okay, so I was thinking, what if you like CRISPR mutate the intron that encodes for miR33?
Uh-huh.
And so then that way, miR33 never gets expressed at the function it needs to be.
Yeah.
And then you just introduce ASO and see if ABCA levels decrease after that.
So you guys are making the same slight error, and that is thinking that ASO decreases ABCA1. It's the opposite. It increases it.
Oh, sorry. Yeah, that's what I meant.
Yeah.
It's
So
through different pathways.
so in a way, so you said silence it, you said knock it out with CRISPR, but it's conceptually it's the same idea. I take out miR33, and then if miR33 is what ASO was using to induce ABCA1, now I should see no effect on ABCA1. Are we good now?
Yeah.
Yeah? All right. Sidi, you going to say something?
Yeah, my answer was kind of saying that we take off miR33, and if ASO is working in association with miR33, then it will have no effect on the level of ABC. But then if it is working independently, then ABC would increase either ways of miR33.
So without miR33, so you all three are saying the same thing. I take out the target of the drug, And if that drug was working through that target and not anything else, then I should see an effect on my readout, right? Everybody agrees? It's very reasonable. There is one small caveat. Yeah, Amelia?
Can we test them both differently, like how ASO binds to MER33 and how ASO targets or affects ABCA1?
So what do you mean by that? Yeah.
Maybe to confirm that that's actually what's going on and like reintroduce MER33 back into the system.
All right.
So you can actually see that that's what's going on.
All right. Okay, so you beat me to the punch. But the idea that we were going with here is if I take out the target of the drug, the pharmacological target, let's call it for a second, then I should see no effect of the drug. If I still see an effect, as Siri was articulating, then it means that it's acting through something else because I took out through CRISPR, through some sort of silencing, I took out the target. So ASO shouldn't work anymore, but if it's still working, it must be through something else. The issue here is how MER33 works. If MER33 inhibits ABCA1, when I take out MER33, I'm already going to be maxing out on ABCA1 expression. So ASO may or may not do anything to increase that expression any further. Does that make sense? So the issue that we bump into is if ASO is inducing ABCA1 expression transcriptionally or post-translationally, then sure. But if ASO is either through MER33 or through some other microRNA, for instance, controlling the translation of ABCA1, I may not see an effect because I already took the break out, so this thing is going to max out no matter what. So conceptually, your idea is perfect. Take out the target of your suspected drug or the drug that you're testing and see if you still see an effect of the drug. In this particular scenario, when we're saying that the drug inhibits an inhibitor, that bumps into some technical difficulties because when you inhibit the target of the drug, you're also inhibiting the inhibitor. So by taking out the target, you're doing the same thing that you're doing with the drug, right? So your experimental manipulation to try and confirm the mechanism of action of the drug and the drug are doing the same thing, and you may have a hard time distinguishing between the two. That's where Alice's idea comes in, and it's perfect. Because if it is true that ASO is working to induce ABCA1 through some totally independent mechanism, through something that has nothing to do with MER33, then adding MER33 should bring the levels of ABCA1 down. On the other hand, if ASO is indeed, this is what I'll do. So temporarily, I'll delete that. If ASO is indeed working through MER33, if I now add MER33, I should be reverting back some of that upregulation. So these are the two potential mechanisms. Adding MER33 here will have an effect. Adding MER33 here won't have an effect. Did that make sense? Because it's almost like if ASO is not working through MER33, adding MER33 cannot bypass this connection. So I should see no effect. I honestly don't necessarily think that this is what happens. I honestly think that, like the author suggests, it does work through MER33. And what we have here is that sort of threshold trigger that at low concentration, the ASO is blocking the function of miR33, at a higher concentration, it actually triggers its destruction. So I do believe that argument. But I just wanted to discuss with you the gymnastics of let's explore other possibilities. Okay?
So for the no effect part, you're saying that if we didn't have miR33, ASO wouldn't have anything to bind to. So ABCA would increase, expression would increase. You can bypass it.
Say that again, because I think you're bringing us back to the idea that we take out miR33 separately, like we mutate the gene with CRISPR or something. The argument is if you take out miR33 and ASO does work through miR33, now you took out the one thing that ASO needs to operate. So you should see that when you add ASO, now there is no increase in ABCA1 expression. Because ASO doesn't mean anything to that cell other than taking out miR33. The issue with that is that if you take out miR33 through CRISPR inhibition or mutation, now ABCA1 levels go up, and it may be hard to see a further increase. So a devil's advocate might say, yeah, but sure, like how can you see upregulation of ABCA1? You're maxed out. Yeah?
I'm just a little confused with, so you're saying that if we remove miR33,
Mm-hmm.
there would be no increase in ASO?
If ASO works through miR33 only, yeah. You should see no increase. Now, no increase may mean it is expressed at normal level and it stays normal level, or it's expressed at 10 times the normal levels, and then when you add ASO, it is still expressed at 10 times the normal level. The scientists who proposed this hypothesis may say, see, no change. So miR33 was the target of ASO. A competitor might say, well, how can you see an increase when you're at 10 times the level of expression? You can't go any higher than that. You can't translate the mRNA more than you already are. Does that make sense? Yeah? One thing that I'd like to point out kind of quickly but emphasize is, I think, Mireya, you started saying, well, I don't know if we could test this idea differently. That's what we do in research labs day in, day out. Think of ways of testing our hypothesis differently, because the more ways that we use, the better. So notice how, like, if that's not the point of this paper, but if this paper, the title of this paper is Mechanism of Action of the ASO, then they would probably have done all of these experiments. Let's take out miR33, see what happens. Let's put in more miR33, see what happens, and so forth. Let's look at the binding. I think you started saying, let's look at the binding of ASO and miR33, maybe the kinetics of that. We might see that at low doses, there's a lot of binding, and then at higher doses, it's where the destruction mechanism kicks in or something like that. Right? Okay, so slight detour. Let's get back on track. And the other thing that they show in this paper is that ABCA1 also regulates astrocytic ApoE secretion. So far we were looking at how ABCA1 lipidates ApoE. That's how we introduce ABCA1 in the context of Alzheimer's, right? So how ABCA1 lipidated ApoE, and lipidation of ApoE can lead to oligomerization of the amyloid beta peptides and so forth. But what this paper says is, check it out, ABCA1, that if you guys remember, in addition to being a lipidase, is also an efflux pump that takes out lipids from cells. So ABCA1 apparently is not only lipidating ApoE molecules on the outside, but it's also putting them there in the first place. So it's secreting ApoE. So because they knew that, they expected that when they treated cells with the miR33 ASO, ABCA1 expression should go up, as they had shown a couple times now, and so should the secretion of ApoE. And what they show us in this experiment is that here's your control ASO, so some scrambled oligonucleotide with no distinct target sequence. Here's the MER33 ASO. The reason you have 2 lanes in each, that's best practices when doing Western blots. Often you load 2 lanes in case that there may be slight differences in loading in each of the lanes. So you often see in papers that things are presented in what appear to be duplicates. It's just a safeguard mechanism for the researchers in case there was some pipetting error or some calculation error and they messed up in one of the lanes. The next one might be better, right? So in this case, they're identical to one another nearly. And what they see is 2 things. And I know that it's kind of hard to appreciate, but they see that when they add the MER33 ASO, presumably ABCA1 expression is increased, there's 2 things that happen. There is a little less ApoE inside of the cells, so that's why we call this the cell lysate. That's breaking the cells up and seeing what's in them. So there is a little less ApoE inside of the cells, and there is a little more ApoE outside. Okay? Again, you can't ever really base all of your conclusions on one gel image. So what they do is they run these things several times, and they do some densitometry to look at the darkness of those bands, and they quantify that. And again, they show that there is a significant increase in the ApoE that is secreted into the medium, and a significant decrease in the ApoE that is found inside of cells. Okay? So, you inhibit MER33, ABCA1 expression is increased, and with that, you expect to see some secretion of ApoE, and that's what happens. I had a question there, what's missing from the secretion measurements? But I'm afraid that we're going to run out of time, so let me address it super quickly. So for the cell lysate, we have the protein that we're interested in measuring, ApoE. And what was tubulin? Do you guys remember? The housekeeping gene, right? The loading control. What tells us that any differences that we see in our protein of interest cannot be easily explained by small mistakes in loading these gels, that we've loaded same amount of total protein in all lanes, and that any differences that we see in a protein of interest is due to differences in the actual levels of those proteins within our sample. What's missing from the secretion measurement is there is no housekeeping secreted protein here. Those are hard to come by, right? So someone who really doesn't like these people might say, well, but how do you know that this more ApoE in the medium is not due to the fact that you collected the medium more carefully, or that that medium was evaporated, so all proteins were a little more concentrated, or something along those lines. So I don't buy this upregulation. Someone who's very, very nasty and nitpicky might say something like that. But I should tell you that for when you're looking at cellular lysates, the argument of housekeeping genes is very easy to tackle. So you use tubulin, you use GAPDH, you use ribosomal proteins. That's fine. When it comes to secreted proteins, there is no, it's rare for cells to have like a housekeeping secreted protein that are going to be secreted at the same level, same amount, under all conditions. That's not something that happens all that often. Okay? All right. Next, they say we wanted to determine if MER33ASO delivered directly into the brain can increase ABCA1 expression in vivo. Mind you, there had been some oligonucleotides against MER33 injected in mice before, but those previous experiments looked at ABC1 expression and cholesterol transport in other organs, liver and kidney, I think. These people want to see, can we deliver the MER33 ASO to the brain and see changes in the brain? Because that's ultimately what matters to anyone doing Alzheimer's research. And I'll go very quickly through this. The answer is yes. and they tried two different approaches. One is to hook a continuous pump to the skull of these experimental mice. So this pump would be periodically deliver through a microsyringe, deliver these MER33 ASO into the hippocampus, or actually one of the ventricles. And then they would measure ABCA1 expression in both cortex and hippocampus. And they noticed that the more they injected daily, the more expression of ABCA1 there was in both cortex and hippocampus. So that was telling them, I'm injecting this thing in the ventricles, these empty hollow spaces inside of the brain, and even then I can see changes in the expression of ABCA1 in the hippocampus, which is an area that is surrounding the ventricles, but even further out in the cortex. When they did a single bolus, it's basically one injection of this thing in the ventricles. When they looked, there was a dose-dependent increase in the expression of ABCA1 that could be detected in both hippocampus and cortex, but apparently the hippocampus was perhaps surprisingly a little less sensitive to a single injection. Right? You would imagine that because it's closer, it's more sensitive, but it was less sensitive. So the hippocampus was not responding to some of the lower doses, whereas the cortex was. Okay, so this is just, I show you this. This is the closing figure in the paper, and it's basically providing a proof of principle that injecting these oligonucleotides against MER33 in brains might work as a therapeutic strategy. So in the discussion, basically they bring up a few key points that they had made before. Pharmacological upregulation of ABCA1 through those, we mentioned them on Monday, those agonists of the transcription factors that normally regulate ABCA1 expression. In other words, drugs that allow for an induction of transcription of the ABCA1 gene. They can be highly beneficial for AD, traumatic brain injury, and atherosclerosis. So this way works, but there is severe systemic metabolic effects associated with their long-term use because the LXR transcription factor that they were targeting with these agonists, they don't only regulate ABCA1, they regulate other things, and those other things they regulate in the long run were causing these metabolic effects. So that's not good. And then they claim, or they argue that although it sounds invasive, these intrathecal, meaning inside of those ventricles inside of the brain, intrathecal delivery is already employed for analgesics and anti-inflammatory agents, so it's not like we're proposing anything preposterous. Our findings demonstrate that ASOs are able to enter neuronal and glial cells without the need for viral and lipid carriers, so there's no need to, you can simply inject it in the ventricles and the ASOs will find their way. There is no need to help them out with viruses or liposomes or anything like that. So that reduces the risk of carrier-mediated inflammatory response. So the whole message of the paper is increasing ABCA1 is good because you will make more ApoE and secrete more ApoE, and more ApoE around neurons and glia are good for scavenging cholesterol and amyloid beta peptides. And so how do we increase ABCA1? Using those LXR agonists was a good idea, but it has long-term effects. Let's use these ASOs that we can inject directly into the brain. So that's sort of the message in the paper. Now, I kind of introduced this idea on Monday. That's great. That sounds fantastic. The problem is that it's a little counterintuitive or a little contradictory with what we had seen in some of those reviews that we discussed when we were introducing Alzheimer's. Based on last week's reviews, we had said that the more ABCA1 you have in the system, the more lipidation of ApoE there should be, and therefore the less non-lipidated ApoE there would be. and that lipidated ApoE induces oligomerization of amyloid beta peptides, whereas free ApoE or non-lipidated ApoE reduces that. So the whole idea was, based on last week, is that ABCA1 was one way or another inducing oligomerization of amyloid beta, either by activating an activator or by inhibiting an inhibitor. Notice that anytime that you activate an activator, you're activating, and anytime that you're inhibiting an inhibitor, you're also activating, right? So that was based on the reviews. This paper basically says, no, having more ABCA1 is a good thing. It's a good thing. So where is that contradiction? So they have a reference to a paper that had been published a few years before in Neuron. Neuron is one of the top 5 journals in neurobiology. And in this paper, they were saying ApoE promotes the proteolytic degradation of the amyloid beta, right? So in a way, it emphasizing the fact that ApoE can inhibit the formation of the amyloid beta oligomers. And in what that paper they showed is that, yes, increase in ABCA1 expression leads to, kind of hard to appreciate here, but they claim that there is more ApoE and there is less amyloid beta. Right? So all along this idea that when you have increased ABC1, there's going to be decreased amyloid beta oligo. So this evidence kind of breaks this idea that ABCA1 promotes the oligomerization of the amyloid beta oligomers. What this is saying is actually the more ABCA1 you have, the less amyloid beta you'll have. I didn't mention it, but that GW is that famous LXR agonist. They didn't do anything with MER33 in this paper. In this paper, they induced ABCA1 expression the old way with the LXR agonist. That's what that GW is. It's the name of the drug that induces expression of ABCA1. These images down here show that when you add that drug, and therefore you increase ABCA1 expression, and therefore you increase ApoE and presumably decrease amyloid beta. In a control mouse, you have no amyloid beta plaques. In a transgenic control, meaning a transgenic animal that is that has a natural propensity, it's one of these AD models. These mice, we call them AD mice. So these are mice that have a genetic propensity to developing Alzheimer's disease because they express an APP mutant that is normally degraded in a way that contributes to the bad amyloid beta, the 40 and the 42, right? So these mice, you'll see references to TG here and in the paper that we'll discuss later, that stands for transgenic, and that is because someone put this mutant APP into these mice, knowing that these mice would be more, that they would have an increased propensity to developing AD, right? So when you take those mice and you don't do anything to them, they develop the beta plaques everywhere, amyloid beta plaques everywhere. But if you give them the drug, the GW, and presumably you induce ABC1 expression and reduce amyloid beta deposition, no plaques. Okay? And one interesting experiment that they did is they say, okay, fine, let's say that you give this drug and that you're increasing ABCA1 expression, and that's what's preventing the formation of these amyloid beta plaques. How do I know, again, we go back to this concept of how do I know that the drug is operating through what I think operates? Right? The idea is I give this GW drug, I induce the activity of LXR. I already told you LXR has many targets. ABCA1 is not the only one. So how do I know that it is really the increase in ABCA1 that is doing the trick. And drum roll, please, your idea, guys. What we do is we take out the putative target and we want to see if the drug still has an effect. So what they did is they gave this drug to mice that are transgenic for developing AD, but that are also wild type for ABCA1, heterozygote mutant, or homozygote mutant for ABCA1. So in this last one, they essentially took out ABCA1, not through CRISPR, through older methodology. This was 2008, CRISPR wasn't even around, but they took out ABCA1. And now what they see is that the amyloid beta is still there 24 hours after, I think this was 24 hours given the drug. So you give the drug, you expect amyloid beta to disappear like it disappeared in the wild type and in the heterozygote, but in the homozygote mutant for ABCA1 is still there. So these guys in 2008 did what you guys were proposing just now. If you think a drug acts through an intermediate target, take that target out and see if the drug still works or not. And in this case, it did not work. Yeah? It came full circle. Not bad. All right, so super quickly, so essentially the paper that we just discussed, the MIR33 paper says, hey, the MIR33, not only will it increase the levels of ApoE, there's also going to be more secretion. The 2008 paper was saying the more ApoE you have in the brain, the less amyloid beta and the less amyloid beta plaques you see in those brains. So having a lot of ABCA1 is a good thing. So I made the executive decision of revising our developing network to indicate that we're still going to go with the fact that ABCA1 lipidates ApoE, and now we're going to go with lipidated ApoE may somehow inhibit the formation of these amyloid beta oligomers. And the one thing that we did was change this from an inhibitory arrow to an inducing one, because paper after paper shows that when you have more ABCA1, you actually have more ApoE. So that idea, I should indicate something that is very, 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 very important. The inhibitory arrow that we had here before, I put it there. Because I said, well, if ABCA1 is lipidating ApoE, the more ABCA1, the more lipidated ApoE you will have, and the less unlipidated ApoE you will have. So I added that inhibitory arrow. But now I need to zip it based on evidence and say, no, okay, having more ABCA1 somehow is lipidating ApoE, but it's also inducing the expression of ApoE and secretion of ApoE. So both things can be true. Yeah.
What if, like, there was, I don't know how I'm going to learn this, but if you have, what if we can't prove that, because they just knocked out the ABCA gene, right? What if the lipidated ApoE still does induce the AD plaques, but there's just so much more ApoE to clean up the mess that doesn't matter how much ApoE there is.
I think what you're saying is, so now these two branches are a little different. Before we said the two branches contributed to AB oligomerization, right? Now what you're saying is, well, hold on a second. Now this branch says inhibit, because I'm inducing an inhibitor. And this branch here says, again, inhibit. Right? What you're saying is, if I take out ABCA1, I'm a little confused. So what you're saying is, I don't have any lipidated ApoE anymore?
I'm saying that does taking out ABCA1 prove that lipidated ApoE reduces ApoE, or is it that maybe there's just so much more ApoE being produced that it just negates the effect of the lipoA4.
It could be, the truth is, if it is true that ABCA1 controls ApoE levels and secretion and lipidation of ApoE, if you take out ABCA1, both things will go down. So this argument that there is so much of this, I would argue, well, but if you don't have ABCA1, you're not going to have that much of it, right? Though I think that argument would break, and here's how. If we take out ABCA1, I suspect that ApoE levels won't change that much. I suspect that ApoE is not controlled by ABCA1 only, whereas lipidation may go down dramatically, because lipidation is an enzymatic reaction that requires that enzyme to substrate specificity that is a lot less amenable to having a wide range. So I can't really imagine how there can be a wide range of lipidases that can act on ApoE. Does that make sense? Whereas there could be a wide range of transcription factors controlling ApoE. Did that make sense? Yeah? Josh.
So, what about ApoE4?
ApoE4. So ApoE4 keeps coming up, right? What about it? Why'd you ask?
So, if you upregulate ABCA1, is there a likelihood that the ApoE that's being secreted is going to be ApoE4?
E4. Excellent question. Excellent question. So the question, so that we're all on the same page. What if I have more ABCA1, right? I'm putting more ApoE out, and we keep saying that that's a good thing. But what if that ApoE that I'm putting out is actually ApoE4? ApoE4 is the one that, because of that mutation and being more prone to being proteolytically cleaved, is actually worse for oligomerization. I would completely agree. If a patient comes to the office, we do 23andMe, even though they went bankrupt. What do people do these days? Ancestry? I don't know, whatever, right? So we genotype them, and they're ApoE4 either heads or homozygotes. I would say do not give them the MIR33 ASO. Because all we're gonna do is pump out more of that ApoE4 into the intracellular space. Does that make sense? I didn't, on purpose, I resist plugging E4 into this network. Here's the reason why. I'm trying to make this network as universal as we can. Not everybody has the E4 allele, right? It's 15% prevalence in the human population, so you do the math, and that means that a smaller proportion of humans carry either one or two copies of the E4. So I don't want to plug E4 here and make it sound like this network applies to everybody when in reality applies to a small set of cases. But your question still stands. So what if ABCA1 is actually pumping out E4? Yeah, that would be really bad. That would be really bad. Here's where I know you're slightly poking your head in the concept of personalized medicine, right? When people say personalized medicine, this is what we're talking about. It used to be like complete fantasy, but these days you have either a family history because you have the E4, or starting to show signs of cognitive impairment, and then you go to the office and they can genotype you quickly. And if they find out that you have E4, chances are they're not going to do anything to increase the levels of ApoE expression because they're going to be increasing the levels of the bad thing. Yep. All right. So last week we had this network. If you go to the network today, you'll see that a few things changed. So there was a change to these axes here. Now the literature convinced me that lipidated ApoE actually can inhibit the formation of the oligomers. It is probably when the levels of lipidated ApoE are excessively high that it becomes inducive. But under normal circumstances, chances are that lipidated ApoE is actually inhibiting, as indicated here. And then the literature convinced me that ABCA1 is good because it promotes ApoE in general, both expression and secretion, and that will lead to clearance of the peptides and therefore inhibiting the formation of the oligomers. Okay? So a slight tweak to the network there. All right, so the rest of today I wanted to discuss quickly the other paper. We'll see how far we get. The other paper that I wanted to discuss this week is CEBP beta is a key transcription factor for ApoE and preferentially mediates ApoE4 expression in Alzheimer's disease. Based on everything that I just said, anything in this paper related to E4 I kind of ignore, right? But I think there was quite a few things that were very important for the relationship between CEBP beta and ApoE in general. So in the introduction, again, ApoE mediates transport of cholesterol and other lipids into neurons through ApoE receptors to make and maintain synapses. Again, ApoE equals good. But ApoE is also needed for amyloid beta plaque formation. There is ApoE inside of plaques, and fewer plaques form in 80 mice models that are also ApoE knockouts. So in a way, this tells you that ApoE is generally good for clearing the amyloid beta peptides, but at some point something may tip that balance, that scale, and it may turn into something bad, because when you actually look inside of the plaques, you do see, biochemically, you see the amyloid beta peptides, of course, and then you see ApoE right there in the middle of it all, right? So that's an indication that it must be playing some sort of role in the formation of these plaques. So ApoE may sometimes not be so good. Okay, so that's the intro. In the intro, they also mentioned that CEBP is a transcription factor that is activated by inflammation in other tissues. So you see CEBP expression go up due to inflammatory signals in macrophages, in liver. And another observation, it's just an observation for now, is that CEBP beta expression is enhanced in 80 patients because, and this is something that others had shown previously, amyloid beta stimulates CEBP beta activation in glial cells. So the idea here is, I just told you that CEBP responds to inflammation in other tissues. We know from last week that the formation of these oligomers can be inflammatory inside of the brain. The brain detects the presence of these oligomers, and glial cells in particular say, whoa, this is not good. We need to clear this out, and then they start producing inflammatory cytokines. And apparently, some of those inflammatory cytokines may be turning on expression of CEBP beta in the brain as well. So CEBP beta is responding to inflammatory signals both outside of the brain and inside of the brain. And an epidemiological observation is that brain CEBP expression increases with age, just like the risk for Alzheimer's disease. So apparently there was one previous study by the same group that had shown a correlation between CEBP beta levels and ApoE levels. So these people in this paper decide to put 2 and 2 together and investigate in this potential relationship a little more closely. The rationale behind this work is as follows. CEBP beta shows a bunch of positive correlations with progression of Alzheimer's disease. And then on the other hand, we know that ApoE can be good, but can also turn into bad, right? So could it be that CEBP beta turns it into something bad? That's what they decide to explore in this paper. So they start by looking at the promoter region of the ApoE gene, looking bioinformatically for binding sites of known transcription factors. They find a bunch of them, CEBP beta being one of them. And what they do in the first panel of the paper is they take neurons, and then they subject those neurons, they incubate those neurons with siRNAs against all of those transcription factors. The idea behind this experiment is I know that all of these transcription factors could bind here. That's what computers tell me. let's see if I inhibit these factors, can I see any changes in the expression of ApoE? Right? So they subject the cells in culture to the various siRNAs, and when they treat the cells with siRNA against CEBP beta, expression of ApoE goes down. With all of the other siRNAs, no changes in expression. That's quantified down here through densitometry, and you see that there is a slight decrease with an siRNA against CEBP alpha, so a closely related transcription factor, so there may be some cross-reactivity there, but clearly a very drastic decrease when the cells are incubated with an inhibitor of CEBP beta. Okay? So yes, any of those transcription factors could have regulated the expression of ApoE. It seems like CEBP beta is the real one. So they see that in neurons and in astrocytes. They also take astrocytes in culture and they treat them with SH CEBP beta. SI and SH, you can think of them as synonyms. SI stands for short interfering, and that is a short RNA molecule, single-stranded, that you dump into the cells. SH stands for short hairpins. It's when people, instead of incubating the cells with a single-stranded small RNA, they incubate them with these little short hairpins. Those things will go into cells, and then dicer will take it up from there and do the rest. Functionally speaking, same thing. siRNA and shRNA, essentially ways of inhibiting your target of interest. So when they take these astrocytes and treat them with an SH against CEBP, I'm not going to say beta anymore, it's a mouthful, so CEBP, the expression of ApoE mRNA goes down, but there was a blot as well to show that the protein went down as well. Okay, so less CEBP, less expression of ApoE. And conversely, when they induce or when they overexpress CEBP in these cells, so they force more expression of CEBP in these astrocytes, now boom, more ApoE. It makes sense. If CEBP is controlling the promoter for ApoE, you take it out, less expression. You put more in, more expression. Yeah.
So, CEBP inflammation triggers more ApoE, which is good for brain and vascular growth, right?
Well, so inflammation might be the one thing that tips this system over from the good to the bad. If there's enough, remember how last week through the reviews we said that one of the issues with Alzheimer's, historically we thought it's the plaques, it's the plaques, it's the plaques. Now we know a little better. It's actually the inflammation that makes the neurons sick and not work very well, right? So inflammation, for all we know, could be the one thing that tips ApoE from being good to being bad. We'll see. But you made that connection. That's important. Okay, so if it is true that CBP is so important for ApoE expression, then if we knock it out, if we take it out from cells, we should see less ApoE expression, right? All of this was cells in culture with either siRNAs or short hairpins dumped onto them. That's very artificial. So let's take a mouse that has 2 copies of the CEBP gene, only 1 copy or no copies. And let's take a look at ApoE expression. And when they do that, they see this appears a Western blot for CEBP itself. Sure, the wild-type mouse has a lot, the heterozygote has less, about half or so, and the knockout has nothing. And then they look at ApoE, and sure and behold, lots of ApoE in the wild-type, less in the heterozygote, no ApoE in the knockout. And down here, actin is the housekeeping gene, similar to tubulin, that we use as a loading control, and basically tells us there was protein in those lanes, don't worry. Like we did load protein in those lanes. The fact that you don't see any ApoE or CEBP is because there was none. Okay? So that's the quantification of that. Now, this is in wild-type mice, or otherwise wild-type, that simply don't have 1 or 2 copies of the CEBP gene. What about these transgenic mice that are prone to developing Alzheimer's? Maybe when you have these transgenic mice with the mutant APP that produces a lot of amyloid beta, who knows, maybe some other CEBP independent mechanism kicks in that controls ApoE expression. Maybe in an AD mouse, something different happens. So they decide to look at that, and there is a way that you can take these transgenic mice and recombine into them the 1 copy or 2 copies or no copies of the CEBP. You just need to know a little Mendelian genetics and it can be done. Right? Not that hard. So when they do that, here's again our friend, the TG. TG stands for transgenic, and it's these mice that are prone to developing Alzheimer's because they express a mutant APP. So in those mice, 1 copy of CEBP, a lot of ApoE. Sorry, 2 copies. 1 copy of CEBP, still quite a bit of ApoE. No copies of CEBP, even in those mice, there is no ApoE. So CEBP is absolutely required for ApoE expression in either a wild-type mouse or even some of these AD mice. Okay? And the quantification here. And last but not least, remember how in cells they had the siRNAs or the shRNAs to reduce the expression of CEBP, and then they had experiments in which they had overexpressed CEBP. They decided to do the same in a mouse. These are not cells in culture anymore. This is a mouse. They used a virus that will introduce the gene for CEBP into neurons and glia and force high levels of expression of CEBP. So in those mice, when they stain for CEBP expression, they see a lot of CEBP everywhere, as expected. That's what you're doing. You're putting a lot of CEBP in these brains. But when they looked at ApoE, again, they also see an increase of ApoE inside of these cells. So both in cells that are growing in culture and in whole animals, there is this dual connection between CEBP and ApoE expression. It is required for it, and it's also, we say, sufficient. If you put more CEBP in these cells in culture or in mice, there will be more ApoE expression. They take it a step further. They say, okay, now that we have these mice that express a lot of CEBP, and in a way, Arshia, addressing your point, we're going to have more CEBP. That's good. Can we see any of the signs of Alzheimer's, right? Because remember, the epidemiology said that there was increase in CEBP expression with age, and there was a correlation between levels of CEBP and what may be I'm gonna say it this way, morbid levels of ApoE, meaning levels that are not so healthy anymore, right? So if that is true, can we see any of that when we simply put in more CEBP? No inflammation, no amyloid beta peptides triggering this whole cascade, simply putting more CEBP. So that's what they do. They take those mice that overexpress CEBP, and after a while they see plaques that you can see easily by staining for the amyloid beta peptide, and they see the famous NFTs, the neurofibrillary tangles that form inside of neurons because tau is hyperphosphorylated and starts multimerizing. And at the bottom here, nu N is a marker that will stain living or healthy neurons. And what they see is that there is a drastic reduction in the number of healthy neurons within these brains. So they see the plaques, they see the NFTs, they see fewer neurons or fewer healthy neurons. That's all the phenotypes related to Alzheimer's, right? Cool, so CEBP beta overexpression in a mouse model of AD enhances the AD phenotypes, meaning the plaque formation and the NFTs. So we go back to this idea of, all right, what is CEBP acting through, right? And their hypothesis was that maybe CEBP is acting through bumping up the expression of ApoE, tipping the balance from good to bad. So that's the hypothesis. So the way that they decide to test the hypothesis is taking out that intermediate. So in this experiment, what they do is they have, all of this is in mice, right? So they have mice that are prone to developing Alzheimer's. Sometimes you use these mice because you want to see something happen. If you do all of this in a normal mouse, the mouse may never develop it or it may develop it in months and you need answers now. So they did all of it in a background that is prone for Alzheimer's. And what they did is the very first lane is nothing. Just these mice with nothing done to them. There is some ApoE. There is a faint band there, I suppose. I mean, these mice still express ApoE. Right? Now, if to those mice you force the expression of CEBP, now there is a lot of ApoE, right? If you give them CEBP and one of these SH controls, meaning a short hairpin that doesn't really target anything, still a lot of ApoE. But here's the first cool experiment. They took those mice, they gave them CEBP, and this time they also incubated these brains, or they had these brains exposed to short hairpins against ApoE. So CEBP is inducing ApoE expression, but then you go through the back door and take it out with one of these short hairpins. Does that make sense? So I am putting a lot of CEBP. Yes, CEBP is inducing ApoE, but then I go through the back door and take it out. They did it that way, and then in the last lane, they did it with a similar approach, and that is exposing these brains, injecting these mice with antibodies against ApoE. When you inject antibodies against something, it's like the immune system. The immune system will go and block this thing. For the immune system, what they want to block, these antibodies want to block pathogens so that they don't make us sick, right? In this case, this is experimental. You're trying to block ApoE so that you prevent it from doing whatever it does. Lara?
On the last part, when you were saying when they added the CEBP,
Mm-hmm.
and then you went through the back door and took it off. Okay, so you're adding it, you're overexpressing it.
Yes.
So the APO increases.
Yes. But the reason you don't see any here is because you also added a short hairpin to ApoE mRNA. So CEBP is inducing the transcription of ApoE, but then you went through the back door and inhibited the translation.
So how would the effects be? So
How does it affect one?
like, okay, you take that out, so what happens?
We'll see that in the next slide. I'm just setting up the experimental approach.
Is this anti-ApoE under the ban?
Yeah, when they say anti-ApoE here, they refer to the antibody they use for the Western blotting. So in a way, that's telling you what protein you're looking at right now. So here you're looking at ApoE, here you're looking at beta-actin, the housekeeping gene. The anti-ApoE here is the antibody that they injected in the brain. Okay? So in both cases, what they're doing is they're putting a lot of CEBP, but then on the side, taking out the ApoE. Why? Because they want to test that hypothesis that what CEBP does, it does through ApoE. Theoretically, if you are putting CEBP, but then you're taking out ApoE through a back door, then CEBP should do nothing now, right? That's exactly what they see. So they had a whole bunch of panels in the paper. I'm showing you this one, which I think is one of the most shocking ones, this amyloid beta plaque formation. In the control, there is some. Obviously, these are AD mice, so they're going to form some. When you give them CEBP, these plaques get darker and larger. If you give them CEBP and a control shRNA, darker and larger. But if you give them the shRNA against ApoE, they're gone. They're gone. So CEBP that normally causes the phenotypes of Alzheimer now can no longer cause plaque formation if you took out ApoE. These last two, same thing, but with the antibody. This is CEBP without any antibody or like some control antibody, lots of plaques, very large, very dark. You put in the antibody that blocks ApoE, plaques are gone. So both are telling us CEBP can cause AD phenotypes, but it needs ApoE. So CEBP does what it does in an ApoE-dependent manner. Okay? Which leaves us, I think we're going to make it just in time, which leaves us with this. What this paper is saying is CEBP, do I have them? Yeah, I think I have them in the wrong order. Okay, so this is the order that I meant. So what this paper is saying is CEBP increases ApoE, and that leads to formation of the oligomers. Less than a half hour ago, we were saying that ApoE lessens the oligomers. So how can we reconcile both? A half hour ago, we were saying that more ApoE is good. Now we're saying more ApoE is bad. So how come? The answer is in a hidden figure. This was supplementary figure 4. I don't know why they hid it so much. In my humble opinion, it's the key figure in the paper. In that figure, what they do is they say, all right, I'm going to take neurons and glia in culture, and I will bathe them in amyloid beta peptides, 40 and 42. Because remember how we said that all of these may be triggered by these oligos and how they kick in inflammation? You should be able to see that in culture, right? So they took cells in culture, they added the amyloid beta peptides to them, and boom, more CEBP, more ApoE, more production of this AEP. This is one of the proteases that cleaves APP the wrong way and makes more of the 40 and 42. This is one of the bad proteases, right? So more oligos or more peptides of the amyloid beta of the bad kind, 40 and 42, will be inducing all of these things. And again, they do an experiment in which they add the oligos, but then they take out CEBP to see if the effect of the oligos on ApoE and AP were direct or indirect, and it turns out that they're more likely indirect because when they took out CEBP, those things disappeared as well. So that is supporting, all of that is here. I don't want to make you later than you are already. So that is supporting this model that the amyloid beta oligos, likely through inflammatory pathways, are inducing the expression of CEBP, and it is CEBP increase that is leading to the increase in ApoE, right? And what I think we can say now is normal levels of ApoE may be clearing the amyloid beta oligo. May be good, right? Normal levels of ApoE. Now, if for other reasons these things start accumulating, this positive feedback loop kicks in, that the amyloid beta oligos will induce CEBP, which will pathologically increase ApoE, which leads to formation of more oligos and eventually plaques.